Good morning, children. Welcome to the biology class, chapter 6, part 2, molecular basis of inheritance. According to James D. Watson and Francis Crick, a DNA molecule is made up of two polynucleotide chains. So, it is double-stranded. One strand is with the purine bases. Purine bases are adenine and guanine. Other strand is with the pyrimidine bases, that is guanine and cytosine, paired with the hydrogen bonds, and they are complementary base pairing. Purine bases are paired with the pyrimidine bases by hydrogen bonds. Adenine is paired with the thymine by 2 hydrogen bond, and guanine paired with the cytosine by 3 hydrogen bonds, and also they are known as the complementary base pairing. Now we will see that one, how these base pairs or the DNA arranged packaging of DNA molecule inside the nucleus. Taken the distance between two consecutive base pairs is the 0.34 nanometer. If the length of the DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with the distance between two consecutive base pairs, that is 6.6 into 10 raised to 9 base pairs into 0.34 into 10 raised to 9 minus 9 base pairs. It comes out to be approximately 2.2 meters, a length that is far greater than the dimension of a typical nucleus. How is such a long polymer packaged in a cell? If the length of Escherichia coli bacteria DNA is 1.36 millimeter, can you calculate the number of base pairs in this bacteria? Now we will take the example of the packaging of DNA helix in prokaryotes like bacteria. Prokaryotes, you know that one, they don't have the well-defined nucleus. There, the nucleic acids are scattered inside the cell. In prokaryotes, such as E. coli, though do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some proteins that are positively charged in a region termed as nucleoid. Nucleus is not that nucleoid. The DNA in nucleoid is organized in large loops held by proteins. Okay. Now, next we will discuss about the eukaryotes, how the DNA is packaged. This organization is much more complex. There is a set of positively charged basic proteins are called histones. Histones are the proteins present in the eukaryotes DNA. A protein, what is a protein? That are the building blocks of amino acids. A protein acquires charges depending upon the abundance of amino acids residues with charged side chains. Histones are rich in the basic amino acids, that are the lysines and arginines. Both the amino acid residues carry positive charges in their side chains. Histones protein is positive charge and DNA is negatively charged. Histones are organized to form a unit of 8 molecules called as histone octamer. Okay, histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure is called nucleosome. In eukaryotes, the positively charged proteins are wrapped by negatively charged DNA is called nucleosome. The core of the histone nucleosome model proposed by Kornberg and the name nucleosome was term coined by P. Audet and Athens. A typical nucleosome contains 200 base pairs of the DNA helix. Nucleosome constitute the repeating unit of a structure in the nucleus is called chromatin. Okay, what is a chromatin? Chromatin is a thread-like colored bodies present in the nucleus. Nucleosomes are present inside the nucleus in a chromatin are seen as beads on string. The nucleosome in chromatin are seen as beads on string structure when viewed under the electron microscope. Theoretically, how many such beads or the nucleosomes can you imagine are present in a mammalian cell? Eukaryotes, example is mammalian cell. The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at the metaphase stage of cell division to form chromosomes. It is clearly visible, you know that when chromosomes are clearly visible in the metaphase stage of the cell division to form the chromosomes. The packaging of chromatin at a higher level requires additional set of proteins, that is not the histone proteins, additional set of proteins that are collectively called non-histone chromosomal proteins. In a typical nucleus, some region of chromatin are loosely packed, stains light, and are active in transcription are known as the euchromatin that are 30 to 80 nanometer in. The chromatin that is more densely packed, thick, they are packed and stains dark, whereas the heterochromatin is inactive transcriptionally is called heterochromatin that is present 300 nanometer. Thank you.